name is Pierce Strayer, and the topic I'm going to be presenting on today is the life cycle of the ergot fungus, or its scientific name, Claviceps puparae. The disease it is most commonly associated with is referred to as the ergot of rye. So the project I made was generated using common art supplies, things such as cardboard tubes, colored felt, modeling clay, pipe cleaners, and plastic insect, and that's what I'm going to be using to demonstrate the life cycle of this fungus. So getting into it, we're going to begin with the sclerotium right here. This is going to be located dormant on the ground during the winter, and this hardened black exterior is going to serve to protect the sensitive inner mycelium from the cold environments with which this fungus is found. So if you look inside the sclerotium here, you can see the sensitive white mycelium located inside that is being protected. As the climate begins to warm and the winter begins to end, this mycelium is going to begin to germinate and produce fruiting bodies known as stoma, represented here by these mushroom-like structures. These are then going to produce ascospores, which are these little white beads here, and those are going to serve as the sexual reproductive mechanisms for this. They're going to be easily dispersed by either the wind or the insects. Now, as the winter is ending and the warmer months are beginning and the germination of the sclerotium begins, plants such as barley and rye, cereal grains, are going to begin the flowering process. So here, this brown pipe cleaner is depicting the stigma of one of these plants, and as you can see, it's closed and inaccessible. However, germination of the sclerotium will occur about 1 week before this process occurs, in which the stigma opens up, allowing access to the ovary, which is represented by this red pipe cleaner here. Now, what this would traditionally allow is the feather-like stigma to trap a pollen grain from the air and bring it down into the ovary for fertilization. However, what is happening is that instead of catching a pollen grain, it's catching one of these ascospores. Once the stigma has caught an ascospore, the ascospore itself will begin to germinate a hyphae, which will extend down into the ovary and begin to colonize that plant structure. The goal of this is to reach the vascular bundle, which is typically going to provide the seed with nutrients and water. And it is linked to the xylem and the phloem, which are represented right here by these orange and blue stripes. So once it reaches that vascular bundle, it will then be supported by the nutrients of the plant itself, and it can continue to grow even more. So as this aforementioned growth continues due to support of the xylem and the phloem from this vascular bundle, the mycelium will expand throughout the ovary and begin to colonize even more, and eventually it will begin to produce a different kind of spores known as conidia. These conidia will then be mixed with the host plant's own natural sugars and sap, and it will form this yellow stripe here, which is referred to as honeydew. And this honeydew is going to be very attractive to insects. So typically in this native host range, these insects would be things such as flies and moths. For the purposes of this, we will be using a beetle. This beetle is going to come, try to feed on this honeydew, and like many other pollinators do, as it's feeding, it's going to get the substance attached to it, as you can see on the underside here. What this means is that as this beetle goes and continues to feed on other plants, it is going to carry those conidia onto those plants and continue the infection process. What this means is that the conidia are going to serve as the secondary inoculum and would be asexual spores. So, as the winter begins to come on again, and this would be about the fall period here, the mycelium will stop growing and it will begin to form this hardened black exterior, reconverting back to the sclerotium we saw earlier, and it will be attached to the plant, where it will either fall off or be harvested by humans. And that harvesting is where problems begin to arise and why this disease is such a problem. These ergots and sclerotiums produce alkaloid compounds, the most famous of which would be lysergic acid, which is a precursor of the drug LSD, and as such causes hallucinations, mass hysteria, and its raw form, the lysergic acid, and it can even be fatal to both humans and livestock. So not only is this fungus rendering the initial barley or rye plant unable to reproduce itself by taking over where the ovary where the seed would form, but it is also rendering the crop unusable for the uses of humans and livestock, and it is just a negative impact economically and ecologically, as well for the health of organisms all around. And thank you.